Hello and welcome to the Lancet webinar on the genetics of Parkinson's disease. Thank you very much for joining us today. I am Elena Becker-Barroso, the editor of the Lancet Neurology, and I will moderate the panel discussion for this event today. But before we start, let me go through some housekeeping points. First of all, most important one is that you can submit your questions throughout the webinar using the Ask the Question box on your dashboard. And then, once the event has ended, you will receive a link to the webinar, and also, once the webinar ends, you will get a survey to complete. Now, this is the agenda for today. And without further ado, we will now listen to Kumsal Bayazit, the CEO of Elsevier, who will introduce our distinguished speakers and panelists.
Hello, and welcome to today's Lancet webinar, which is being broadcast worldwide. I'm Kumsal Bayazit, CEO of Elsevier, a global information and analytics company serving research and healthcare communities and proud publisher of the Lancet portfolio of journals. My Lancet colleagues and I are delighted to introduce Professor Thomas Gesser, today's guest presenter and an expert on the molecular basis of Parkinson's disease. This severe progressive disease affects around 8 million people worldwide, and around 10% of these patients have a genetic predisposition. Thomas is a world-leading specialist in this field who has been researching genetics of Parkinson's disease for many years at the Hertie Institute for Clinical Brain Research at the Medical Faculty of University of Tübingen. Together with Andrew Singleton and Ellen Sidransky from the National Institutes of Health in the US, Thomas was recently awarded the 2024 Breakthrough Prize in Life Sciences for identifying risk genes for Parkinson's disease. The Lancet Group and I extend our warmest congratulations to Thomas, Andrew, and Ellen. We are grateful for Thomas's time and expertise, which he has generously agreed to share today. I hope everyone enjoys today's Lancet webinar. Thomas's engaging presentation will be followed by a lively discussion with Tanya Simuni, Arthur C. Nielsen Jr. Professor of Neurology and Director of the Parkinson's Disease and Movement Disorder Center at Northwestern University Feinberg School of Medicine in the United States, and Christine Klein, Professor of Neurology and Neurogenetics at University of Lübeck and University Hospital Schleswig-Holstein in Germany. Thomas, thank you very much for coming. We're excited about what you're going to present to us today, and the floor is yours.
Thank you very much. And from me also a warm welcome. Good afternoon, good morning, good evening, wherever you are in the world. Here are my potential conflicts of interest for you to read. And I'll go right into trying to explain the current status of the genetics of Parkinson's disease, which has finally come to a stage that really allow us to develop novel and personalized stratified treatments for the patients that actually modify the disease course. At least that is what we hope. So when James Parkinson described the disease more than 200 years ago that today bears his name, he very clearly elucidated the clinical motor syndrome. He said involuntary tremulous motion, you just could see that on the video, involuntary tremulous motion with lessened muscular power in parts not in action and even when supported, with a propensity to bend the trunk forward and to pass from a walking to a running pace, the senses and intellect being uninjured. Now, this last half sentence is something that we actually disagree at the moment. He described the motor syndrome very well. We can treat the motor syndrome very well. You just saw this on the other slide, where the patient has come into an on state, but we cannot do much about the progression of the disease and about the cognitive symptoms. The disease was researched for over 100 years now, and almost 100 years, the pathology was described. The degeneration of neurons in the midbrain, in the substantia nigra, which you can see here on the left-hand side. And about at the same time, Friedrich Levy, a German neuropathologist, described the characteristic inclusion bodies that are found in the remaining surviving neurons of a Parkinson's disease patient, and they are called the Lewy bodies or Levy bodies. At that time, nobody knew what those Levy bodies consisted of. The neurochemistry of Parkinson's disease was described in the mid of the 20th century based on animal experiments and the crucial role of dopamine, dihydroxyphenylalanine, was elucidated by Arvid Carlsson, who obtained the Nobel Prize for that in 2000. And what you can see here on these nuclear medicine imaging slides is the degeneration and the lack of dopamine, either by levodopa PET or by a DAT-SPECT, which is a dopamine transporter SPECT imaging. So there is a loss of dopamine in the basal ganglia, which is the consequence of the degeneration of the dopaminergic neurons in the substantia nigra. The real cause of this neurodegeneration was unknown and is still in many cases not really totally understood. But in the 80s, it was generally considered to be a toxic disease. This was based on the discovery by Bill Langston that a compound MPTP, a contaminant of illicit opioid production, causes a syndrome that is very similar to Parkinson's disease. Around the same time, however, a family was identified originating from southern Italy, the so-called Contursi kindred, in whom Parkinson's disease, or a disease that very much looked like Parkinson's disease, was inherited in a very clear autosomal dominant fashion from generation to generation. You can see all the dark symbols are the affecteds in this family. This was a fairly severe form of Parkinson's disease with an onset relatively early, average 44 years, rapid progression, and dementia. Now, this type of family was at that time necessary to identify the genetic underpinnings of a disease. And this is what Bob Nussbaum at the NIH did. And he identified in this particular family a mutation in a gene that is called alpha-synuclein, or SNCA, as causative. And what he found was a point mutation, a substitution of the alanine in position 53 by a threonine. So a single point mutation was able to cause the severe form of Parkinson's disease in this particular family. Now, what was interesting is that this discovery of alpha-synuclein very soon led to another discovery, namely that the Lewy bodies that Friedrich Levy described decades before actually mainly consist out of aggregated forms of this protein, of the encoded alpha-synuclein protein. And that was discovered by Maria Grazia Spilentini and Michel Goedert and Russ Jakes in the UK. A few years later, Andy Singleton, my co-recipient of the Breakthrough Award, and Matthew Farrer together found that not only point mutations, but also duplication, and in this particular case, triplication of the alpha-synuclein gene could cause a dominantly inherited form of Parkinson's disease. So it was not even a change in the amino acid sequence, it was simply a dosage effect of the gene that could lead to these aggregated forms of alpha-synuclein and the ensuing neurodegeneration. What is now possible is to mimic or model this aggregation process in vitro. So the formation of alpha-synuclein aggregates from monomers can be basically modeled in vitro by adding a tiny little seed, which you can see here on the right-hand side in this circle. It's the square seeds that are added into a solution of recombinantly produced alpha-synuclein solution. And if you take this mixture and shake it for 2 days approximately, the conformational changes lead to a growth of aggregated alpha-synuclein fibrils that can be visualized by fluorescence. If you simply add thioflavin as a fluorescence detector, you can monitor this process of aggregation in a test tube. This is called a seeding aggregation assay. It's derived from the prion protein field of research, but it also works for alpha-synuclein, and it actually is a very sensitive and highly specific test for the process of alpha-synuclein seeding aggregation in Parkinson's disease patients. It works very well from CSF. It doesn't work quite as well from the blood yet, but I'm pretty sure that we'll get there within the next couple of years. There's one problem. Alpha-synuclein point mutations and multiplications account for only a minute minority of Parkinson's patients. Less than 1% of all patients have those conditions that introduce the alpha-synuclein aggregation and consequently the neuronal degeneration. So there must be many other causes for alpha-synuclein aggregation and neuronal degeneration other than mutations in the alpha-synuclein gene. And in fact, by status 2024, this is a not totally complete list of genes that have been identified over the past years that contribute in one way or another to the neurodegenerative process and induce Parkinson's disease with high penetrance and a strong effect. Those gene mutations that were detected point us to a number of major cellular pathways that have been implicated now in the disease. And I have already mentioned the protein aggregation that was discovered by the mutations and duplications, triplications of the alpha-synuclein gene. But there are also others, and I will go through them in 3 steps in the rest of the talk. There is a mitochondrial problem that is induced by other mutations that have been identified. Those in green here, the recessive mutations in Parkin, PINK1, and DJ1. There is a problem of vesicle sorting and transport, which is probably the effect of mutations in the LRRK2 gene. And there is the lysosomal function or dysfunction that contributes to the pathology of Parkinson's disease in patients with GBA1 mutations. And I'll go through those 3 different pathways to Parkinson's disease, each at a time. The first mutation that was identified after the alpha-synuclein discovery was in very young patients from Japan who developed an autosomal recessive form of Parkinson's disease. Their onset is often in the early 30s or even in the 20s, and 3 mutations for these recessive forms of autosomal recessive forms of juvenile or early onset Parkinson's disease have been identified: Parkin, PINK1, and DJ1. Now, the analysis of those loss of function mutations have pointed us to a different pathway, not protein aggregation, but those genes or the gene products are involved in the mitochondrial quality control, and they control a process called mitophagy, so the removal of damaged mitochondria from the cells. And those patients, therefore, probably have a different molecular class of Parkinson's disease. And if you remember the protein aggregation assay, the synuclein aggregation assay that I described to you just now, the patients with Parkin mutations, at least in most cases, do not have protein aggregation of alpha-synuclein in these synuclein as seeding assays. The next form of Parkinson's disease that I would like to describe briefly is the ones that is linked to the LRRK2 gene mutations. Discovered several years later, in 2004, Andy Singleton and our group identified mutations in this particular gene as causing another dominant form of Parkinson's disease. And you can see here the pedigrees of the 2 papers that were published, not quite as large as the Contruzzi kindred that led to the discovery of alpha-synuclein, but large enough that at least combined, they had enough statistical power to clearly identify mutations in this LRRK2 gene. So the LRRK2 gene apparently causes a much more common form of inherited Parkinson's disease. In Northern Europe, it's still relatively rare. 1 to 2% of cases have LRRK2 mutations. But in Southern Europe, it's already getting more common. And particularly common is mutations in the LRRK2 gene in Northern Africa, in the Arab Berber population, and in Ashkenazi Jews from Israel. And this is due, both those increased frequencies of mutations are due to founder mutations. One in the Basque population in Spain, that's mutation R1441G, and the G2019S mutation, which is the most common LRRK2 mutations overall, which is particularly common in Ashkenazi Jewish and Berber Arab populations. Those mutations occur both in familial and in sporadic disease, which means that those mutations have reduced penetrance. And here you can see the penetrance estimates from a number of different studies. And you can see one, that the penetrance is clearly age dependent. So it starts at age 50 here now and goes up to age 80. And it, of course, increases with age. But it's also very different in different populations. Average would be here probably around reaching a penetrance of 40, 50% at the age 80. But it varies between 30 and 80 or 90%. And that is probably due to the different genetic backgrounds of those populations. This is a typical LRRK2 patient. It's one of our patients. At the time the video was made, she was age 64, 9 years into her Parkinson's disease, still treated only with pramipexole, with a dopamine agonist, no dyskinesias, no cognitive impairment, with a MoCA of about 29. So that is typical for a LRRK2 patient, mid-age of onset, relatively mild form of Parkinson's disease, well treatable with dopaminergic medication. But the spectrum is very wide, that you can see here in this age of onset description of a couple of hundred of Parkinson's like 2 patients with an average age of onset around 55 to 59, but ranging from 20 to 80. So there must be a number of modifiers that alter the expression of the LRRK2 gene. And one of them is different genetic factors. I already mentioned the genetic background of the population, but there are also a number of genes that have been identified to modify LRRK2 expression. For example, the CORO1C gene that was identified by a genome-wide screening of LRRK2 patients with LRRK2 carriers with and without Parkinson's disease. And you can see a clear difference depending on the allele that is present in the CORO1C gene. But there are also other non-genetic modifiers. For example, and that's just one example, non-steroidal anti-inflammatory use may modify the disease risk in Parkinson's patients or in carriers of the LRRK2 mutations with a relative odds ratio of about 1/3. So Parkinson's LRRK2 mutations carriers who take regularly non-steroidal anti-inflammatory agents have a reduced risk to develop actually the disease, which is probably due to the fact that LRRK2 is a gene that is expressed also very strongly in the immune system, and therefore is influencing the process. So LRRK2 mutations, what do they do? The LRRK2 gene is a kinase that phosphorylates another group of signaling proteins, namely Rab proteins. And these proteins are signaling proteins that are GTPases that influence a whole range of molecular mechanisms in the cell. And mutations in the LRRK2 gene lead to an overactivity of the kinase. Therefore, a potential treatment for patients where this pathway is activated could be kinase inhibition of specific LRRK2 kinase inhibitors. And I will come back to that at the end of the talk. There's one peculiar feature of LRRK2-related Parkinson's disease that was already apparent to us during the time when we tried to identify the mutation. That is that patients with LRRK2 mutations can have different pathologies. In this particular family that was also used to identify the gene, there are some patients who have typical brainstem Lewy body pathology. There was one patient that had cortical Lewy bodies corresponding to Lewy body dementia. There was another patient with autopsy who had just extracellular pigment but no Lewy bodies, and there was even one patient with tau pathology. So this is called a pleomorphic pathology, and correspondingly, this has also been seen in cohorts of patients with LRRK2 mutations, where about 1/3 of those patients, now this red square dropped a little bit, it should be up here, the G2019S mutations, where LRRK2, where Lewy bodies are found in about 2/3, 64%, and and 30% do not have Lewy bodies, while with other mutations, it's the other way around. Only 30% have Lewy bodies, 70% do not have Lewy bodies. And this also corresponds to the seeding aggregation assay, which is very sensitive and detects practically all patients with sporadic Parkinson's disease. Here it's 93% sensitivity of positive seeding assays in the sporadic patients, while only about 2/3, 67% of LRRK2 mutation carriers have a positive seeding assay. So the correlation between alpha-synuclein aggregation and Parkinson's disease in LRRK2 mutation carriers is not quite as clear as I mentioned before with the synuclein mutations. Finally, the last group of genetic forms of Parkinson's I would like to describe are those with a GBA1 mutation. GBA1 is a gene that has been well known from neuropediatrics as causing, in a biallelic mutated situation, the known pediatric disease of Gaucher's disease. And it was found out, and Ellen Sidransky found that out, and she is also a recipient of the Breakthrough Prize, that the heterozygous parents of patients with Gaucher disease have an increased risk to develop Parkinson's disease. And she confirmed this with a large international study. In fact, GBA1 mutations, and this is a slide from a recently published large genetic screening study from the ROPAT cohort, the Rostock Parkinson's disease study, with 12,000 individuals genotyped. The GBA variants are the most common strong risk variant for Parkinson's disease, and about 8 to 10% of patients carry that. Different populations with different frequencies, but at least in Western populations, this is the most common gene mutation or variant that carries a Parkinson's disease risk. So what is GBA1 coding for? It's coding glucocerebrosidase, GCase. This is a lysosomal enzyme that is transported through the endoplasmic reticulum to the lysosome, which is also responsible for degrading most of the alpha-synuclein. And in normal cases, the alpha-synuclein is degraded in the lysosome by a mechanism called chaperone-mediated autophagy. But if glucocerebrosidase lacks in the lysosome, its normal function, namely cleaving glycosylceramide to ceramide and glucose, is deficient, lysosomes are dysfunctional, and alpha-synuclein aggregation follows. At least that's a fairly simplified version of what we think is behind the connection of GBA1 and Parkinson's disease. Now, GCase or GBA1 mutated Parkinson's patients have, again, a severe form of Parkinson's disease. They have a more rapid progression, they have common dementia, so they look more like the alpha-synuclein mutated cases I showed you before than like the LARC2 mutated cases, and correspondingly, those patients also have a strong and robust alpha-synuclein seeding in their CSF. So they are in the group of very strong alpha-synuclein pathology Parkinson's patients. Now I have to stop the slide first, and now I can go on. Okay, so now I showed you 4 different types of monogenic forms of Parkinson's. Very early onset, medium onset, late onset, with different severities. They also have different underlying gene mutations, they have different pathologies, and they have different properties in terms of their alpha-synuclein aggregation propensity. So what can we derive from this in terms of stratified treatments? Oh, no, sorry, another paragraph I just forgot. Those were that's just the tip of the iceberg. The 15% of those patients who have strong mutations that drive the disease. The vast majority, 85% of the patients, do not have these genetic strong driver mutations, but they still have a genetic component. And this is being analyzed by so-called genome-wide association assays. So the genome-wide association assays are basically association studies that cover the entire genome, that use normal genetic variability to identify risk variants and protective variants across the entire genome. And that has been done in very large patient cohorts. So here, 37,000 patients and 1.5 million controls. And more than 80 genome-wide significant risk loci have been identified using those studies. Actually, now there are more than 100. And interestingly, the genes that are identified in the families are also found in those genome-wide association studies. So you can see GBA1, alpha-synuclein, SNCA, and the LRRK2 locus, not the recessive disease genes, Parkin and PINK1. So they're only the dominant disease genes that also confer a risk to sporadic Parkinson's disease patients. And this shows us the genetic architecture of Parkinson's disease, which is like for all other or most other complex genetic diseases, a continuum of rare and very rare mutations that carry a high risk to develop disease and common genetic variability that carry an individually low risk, but they act together to promote the disease risk. And then there's everything in between. And in this case, it's probably the LRRK2, G10-19S, and the GBA mutations that are intermediate frequency, intermediate risk variants. Now, what is important is to remember that those variants act in different by different mechanisms. The very rare mutations are coding variants. They change the function of the protein. The common genetic variability acts via non-coding mechanisms, mostly probably via gene expression. So what can we learn from that in terms of treatments? Basically, I would like to pick out two forms, the one with a strong alpha-synuclein aggregation and the one with a milder or less consistent alpha-synuclein aggregation that is caused by LRRK2 mutations. So the ones with the strong alpha-synuclein mutations, one could try to reduce alpha-synuclein as a aggregating protein. And this has been done and tried in the Pasadena study, which has been performed for about a year. Patients have been treated with alpha-synuclein lowering antibodies, and the primary endpoint of the study was not reached. We did not see a difference between the treated and the non-treated group. However, as I just showed you, Parkinson's disease is heterogeneous. So maybe the reason for this failure is that the patient group was not stratified appropriately. In a follow-up paper that was published just recently in Nature Medicine, a post-hoc analysis was done in a subgroup of patients who have a form that is called diffuse malignant form of Parkinson's. So those with a lot of cognitive impairment, with a lot of autonomic disturbances, with a rapid progression. And remember, that is the type of Parkinson's disease that I just described for the GBA mutated cases. So if we would identify this particular subgroup and use it as a treatment group for alpha-synuclein lowering treatment, this might actually be a stratification that leads to a more successful alpha-synuclein lowering treatment, and we just are actually planning that for a GBA cohort. The other potential novel treatment that I would like to point out is those with the LRRK2 mutations or stratified groups that correspond to LRRK2 mutations with LRRK2 inhibitors. Some of those inhibitors are also already in clinical trial. For example, one that has been developed by Denali and Biogen, initially with two arms, one with LRRK2 mutations, one without LRRK2 mutations. But now the LRRK2 mutation arm has been closed, and the ongoing ILLUMA study uses the LRRK2 inhibition, both in early Parkinson's disease sporadic and in LRRK2 mutation carriers. Now, will this be successful? We don't know. The problem might be, again, as in the Pasadena study, that the early PD group is not really stratified for those who have an active LRRK2 pathway. And given the heterogeneity that we saw in the genetic forms of Parkinson's disease, and it is likely also in the sporadic forms, this could be a problem. And so could there be a readout that allows to identify those Parkinson's patients with idiopathic, sporadic Parkinson's disease that will respond to LRRK2 inhibition? Well, there are some. This is just one example. There are patients with what is called a cohesion phenotype, that is a phenotype in peripheral blood cells that is that does show a lack of centrosome formation, and this could be used to isolate those patients, to identify those patients who will respond to LRRK2 inhibition. So, as a conclusion, the genetic evidence now points to different pathways, different pathogenetic pathways that lead to different pathologies with overlapping clinical phenotypes. The phenotypes are determined by interactions between relatively strong driver variants in a minority of cases, but in cooperation with multiple common but weak risk and resilience variants in all patients, plus non-genetic factors, as I mentioned, the non-steroidal anti-inflammatory agents. And those disease-modifying pathways can be used to stratify patients for novel disease-modifying treatments, as I just mentioned them. And with that, I'd like to close, and I thank you all for your attention.
Thank you so much, Thomas. A brilliant, brilliant presentation. It's spectacular how you have managed to condense and give us a summary of over 30 years of work by you and your team and by other great contributors to the field in just about 30 minutes. Yes, in just about 30 minutes, just fantastic, really spectacular. So thank you very much for that. I'm actually delighted today to be in such a good company and of this plethora of great movement disorders specialists. So welcome, Tanya. Hello. Hello there.
Good
Hello,
afternoon.
welcome.
Good afternoon,
Hi.
my pleasure to join.
Hello. And hello, Christine.
Hello, and thank you very much for having me. It's a great pleasure and honor to be part of this.
Fantastic. So before we start with our panel discussion, if you allow me, I would like to I have a couple of questions for Thomas before we move forward. And well, first of all, I would like to remind our audience that they can ask questions as well. So they have in their dashboards that box to submit their questions. But my questions to you, Thomas, are as follows. First of all, I would like to know, for a young individual in her early 20s, for instance, what's her lifetime risk of developing Parkinson's disease? And what is more important, those rare, very penetrant variants that you spoke about, or maybe the common variants with smaller effects?
Okay, so the overall lifetime prevalence, let's say if you reach an age of 65 and older, is about 1 to 2%. 1 to 2% of the population will develop Parkinson's disease by the age of 65 or so. Both components, so now we know that about 15% of those individuals who develop Parkinson's disease have one of those strong driver mutations, 15%, so 85 do not. So overall, for the overall population, it is probably more relevant to identify the multiple common risk variants that interact and that act together to develop the disease, but the rarer strong driver mutations tell us, at least initially, more about the molecular pathways that are involved. and give the clues to where to intervene with treatment.
I see. Thanks for that clarification. Now, I'm really sorry, but I need to ask you this $1 million question, or maybe these days this is a $1 trillion question, which is the one on, of course, selective vulnerability, right? Neuronal selective vulnerability. So, of course, here, well, the big elephant in the room, as I would say, why these people lose the dopaminergic neurons of the substantia nigra. So in other words, what is genetics telling us about that?
Well, that is a very good question, and that has been discussed for decades now without a very clear answer, I have to say. There are probably a number of factors that make dopaminergic neurons particularly vulnerable. One is, for example, that they have a very high metabolic activity, so they fire a lot constitutively. That makes them very vulnerable because they have a strong energy consumption, and that makes them vulnerable at least to the mitochondrial deficits that play a role in many of those forms of Parkinson's disease. They are also very highly arborized neurons. So one dopamine neuron supplies dopamine to hundreds of thousands of neurons in the projection areas, and that makes them special. They also have very long axons that makes them vulnerable. So there are a number of things that I think that make dopamine neurons particularly vulnerable to all kinds of metabolic insults, and that leads to the fact that the dopamine degeneration is the most visible part of Parkinson's disease. But as I mentioned, it's not the only one. You know, patients become cognitively impaired. They have a lot of autonomic disturbances. So it's just the most prominent, most visible, but not the only clinical feature.
I see. So now, unless Tanya or Christine have some burning questions to ask, we can move into our panel discussion. And I would like to focus this discussion on the practical use of genetics in a clinical setting or in a research setting. And at that respect, I believe that our audience will be very interested in knowing your views about whether a clinician should offer genetic screening to every patient. Every neurologist will do that as a routine part of the evaluation of a patient with a movement disorder, or a part of the care trajectory of that patient. What do you think? Thomas, could you perhaps start with your opinion?
Okay, it's also a very good question. I do offer genetic testing to the majority of my patients. I have to say that very often, at least our patients say, well, what is the consequence? What would I benefit from? And then I tell them, well, you could be eligible for specific novel treatments, but your immediate treatment would not change. So at least in my experience, I do offer it generally to all my patients, but many of them say, I'd rather not look so closely into it. I don't know how the others experience that, but I think that this could be something that if we do that, if we make more advertisement for this type of further investigations, that this will change over time.
What's your view about this, Tanya?
So first of all, I want to start with echoing Elena's command, Tomas, of you covering the spectrum over 30 minutes.
Yeah.
I will start addressing the question as a clinical investigator who designs and implements a lot of clinical trials. And there, I think, unequivocally, knowledge of the genetic composition of individual spanning across monogenic variants, as Professor Gasser has outlined, and genetic risk what we frequently refer to genetic risk score, is essential. I think that no one would debate that. As a clinician, I side with Professor Gasser that in my practice, I do offer and counsel the patients regarding the value of genetic testing. However, I want to preface that in a lot of countries, definitely in North America, genetic testing is not covered by the payers. So we are fortunate to have a research program that allows clinically certified genetic testing to be offered to all people with a diagnosis of Parkinson's disease. Similar to what Professor Gasser has indicated, I specifically focus on individuals with newly recently diagnosed disease for the reasons of stratification for clinical trials. But I believe that the way the field is moving, it has to become the standard of care, and certainly would like to hear Christine's perspective.
Christine.
Thank you. Yes,
please.
thank you. Thank you very much. And also from my side, Thomas, thank you very much for this insightful, beautiful presentation. A lot has been said, and Thomas, you also already alluded to the differences in different genetic types, monogenic types of Parkinson's, where sometimes also for counseling issues, for example, it can be very helpful to know the genetic status, such as in a patient with alpha-synuclein Parkinsonism, or even with a GBA1 variant, where, for example, and you showed this, the clinical course and the prognosis, also, for example, the response to deep brain stimulation may be slightly different from that in a person without that type of variant. So I think from that perspective, I can only agree with what everybody has, or what you have already outlined. Maybe one additional perspective. So first of all, I guess, and this is something you said, Elena, so you also mentioned the research perspective, and maybe I can come from that side. And really, the research perspective has grown very, very large. So the Global Parkinson's Genetics Program, or GP2 for short, is currently in the process of testing 250,000 individuals and performing whole genome sequencing in 80,000 of those across the entire globe. And just for the monogenic network, we have already included more than 100 centers really across the world. And so this is where Parkinson's and genetic testing becomes, in a way, although it is not set out as a genetic testing facility, but genetic testing and genetic testing results become available more and more. And so I think we have to consider, on the one hand, the research perspective, and on the other, the clinical perspective. In my own practice, we also offer the research program to everyone. And I have to say, most patients are really interested. Some actually come to us because they know we do research. So it may be a biased population. On the other hand, when it comes to the clinical setting, I would do exactly, Thomas, as you just described. I would offer it to those who I suspect a monogenic cause, or those with an earlier age of onset. And this, of course, and you mentioned this, Tanya, will change, immediately when the gene-specific or I guess gene-specific treatments become available. And hopefully,
thank you very much. Thank you for this. Insights. And before we move on to the research setting in full, I would like to pose some questions that are actually coming up already from our audience, because I think they are very pertinent. And many of them relate with the clinical setting in a particular setting, obviously, because I am talking to you from London, and Thomas, Christine, you are in Germany, and of course Tanya is talking to us from the United States, and obviously we have audience all over the world, and they're asking questions about clinical settings in India, for instance, clinical settings in other parts of Asia. I mean, are there specific populations that merit a specific approach in the clinical setting? And with that question, then after that, we will move into the research setting. Maybe Thomas, you would like to respond to that question?
Yeah, well, obviously I cannot, I think that genetic testing on a clinical basis is very different in different countries and areas and regions of the world. Christine already pointed that out. However, particularly the GP2 program is now really reaching out to globally to all so far underrepresented populations to first do genetic characterization on a research basis. But I think in most of those cases, they're also working towards making this available on a clinical basis. And Christine, maybe you can say a little more about that, because that is basically your field in the GP2 program.
Yes. Thank you very much, Thomas. Yes, I'd be happy to elaborate a little bit further on this. So this is exactly what is happening. So initially at the set out, it was purely a research program, but then it became clear now that, of course, there's a big need. And of course, the data is being fed back to all the investigators, and now they have the data and they want to share it with their patients. And the other big study, Thomas, you kindly mentioned the ROPAD study, the other big study that appeared back to back in the brain issue was the PD generation study that Tanya, I believe you also referred to. And that is a study that was always from the very set out designed to feed back results, as does ROPA, or did ROPA, by the way, to the patients in a semi-research setting, if you will. And within GP2 now, there is, and this effort is now led by Ai Hui Tan from Malaysia, there is now a specific effort to really return research results to investigators, and they then in turn return them to their patients. And there's, as we speak, I actually today got the slides for a webinar that we are going to present within GP2 and make it available to everyone to learn and to make this possible. And the final point really is that PD generation is now already being extended from North America to all of the Americas, so including the South America, for example, and that way clinical grade diagnostic results will be made available to at least a subset of these patients.
Excellent. Thanks very much for that. Then, so it seems to me that you all agree in thinking that in clinical research, in clinical studies of Parkinson's disease, the need for genetics is unquestionable, for the use of genetic findings, genetic testing as a way of stratifying patients. So let's talk a little bit about that, because it seems to me, maybe wrongly, that actually there is this paradoxical situation in which maybe genetics is pretty well defined, whereas phenotypes are not so well defined anymore. So what's going on? Could you please explain to our audience what the situation is at that respect? Perhaps we can start with Thomas.
Okay, yes. It's a good question, and I think it's a very pertinent question. And I think we have to remind ourselves that a genetic mutation has an immediate effect on the gene that it encodes. And this gene has a certain function. For example, like 2 has a kinase function. And so the kinase function will change depending on the mutation. But everything that comes downstream from there is really a complex network of feedback loops that really do not allow a causal one-to-one reasoning. And that's why right now at the Movement Disorders Congress in Philadelphia, one of the major topics was co-pathology. Parkinson's is very simplified terms, a disease of dopamine neurons with alpha-synuclein aggregation. In reality, patients have very different pathologies. There are a number of aggregation-prone proteins that are playing a role, and those are all elderly or old people who have a lot going on in their brain. And therefore, we should make it as simple as possible, but not any simpler. It is a complex disease that is a very, very complex network of causations and feedback loops. And that's what makes the clinical categorization, I think, so much more difficult than just to say, okay, we have one gene, and that's a reasonable answer.
Excellent. So Tanya, what's your views about that? Will you agree with Thomas about that?
I absolutely agree with Thomas, as always, and would take it one step further, right? Parkinson's phenotype is complex. All of us tell every person with Parkinson's, you've seen one person with Parkinson's disease, you've seen one person with Parkinson's disease. But how can we move it further to what all of us strive towards is personalized therapeutic approaches? Genetics are essential because, as Thomas has eloquently outlined, they lead us to specific mechanisms and pathways. Genetics certainly is one part of the complex equation of mechanistic pathways. And in addition to what Thomas has talked about, co-pathology, probably partially driven by co-pathology, but more and more across neurodegenerative diseases, we talk about resilience, right? Why that 100-year-old person who carries genetic variant that their sister or brother have manifested with the clinically manifest disease 40 years younger than them never develops that, right? Resilience, susceptibility. How do we study that mechanistically? Because all of that leads to that next generation therapeutics of taking as many factors into the decision point equation. We are not there yet. It will require combination therapies, but unless we characterize individuals biologically the best we can today and build on that with every new development in the science, because ultimately, in order to characterize living individual, that's what we need to do. We need the biomarkers, right? We have genetic biomarkers, we have sequencing, right? But we need biomarkers of the pathways, we need biomarkers of lysosomal dysfunction, we need biomarkers of mitochondrial dysfunction, inflammatory pathways, all of that leading to that, building that complex network, right? And translating it into informed decision-making about therapeutics. And speaking of the studies, I do want to highlight the Parkinson's Progression Marker Initiative study that truly has led the way in that task, right, and objective of biomarker characterization of individuals across the spectrum, starting from people with newly diagnosed Parkinson's disease, building on the cohort of individuals with monogenic, most common genetic variants, and not common as well. And then expanding further the cohorts into individuals at risk, both at risk based on the clinical features, loss of sense of smell, has very high correlation with the presence of underlying synuclein pathology. As a lot of you know, particular sleep disorder, REM behavior disorder, acting out dreams has very high predictive risk of developing one type of synucleinopathies, or whether that is neuronal synucleinopathy, predominantly glial synucleinopathy. So we need more studies like that. We need structured prospective ascertainment of individuals. So long answer to the question, but that's what the field needs.
Fantastic. So we have tens of questions that obviously you wouldn't have the time to answer. So I'm going to try my best to summarize them in themes, and perhaps a broader one will include many of these questions that we got here from our audience. And they all seem to be very eager to understand when these genetic findings will be available, will be moving to the clinical setting, and what's your view about the future, but kind of sort midterm? It's like, say that you have the crystal ball and you can perhaps speculate. I'm going to ask you actually to speculate about your views for the field in 5 years or 10 years' time. So if you could predict how things will stand in Parkinson's disease with the use of genetics in 5 to 10 years from now, what will be your answer to that, Thomas?
Okay, so I think we have seen a huge progress in the genetics of Parkinson's disease, and I think this will go on and even accelerate. I think this global view on the genetics of Parkinson's disease is really something, a new aspect that accelerates scientific insight to a major degree. And it will not only help to broaden the applicability to other so far underrepresented populations, which is good in itself. You know, why shouldn't people in Africa have the same chance to access genetic information than people in Europe? But it will also help to understand the genetics of European populations much better, because so far much of that is associations. We know which variants are associated with the disease, but we do not really know much about the causal mechanistic connections. And this will be much easier to understand if we can look at it in different populations, because the genetic structure is different, because the environmental influences are different. And if we have this global view, we can much better understand the etiology of the disease also in our European homeland, right? So everybody will benefit, and that, I think, is a great thing, particularly in these days today.
Right. What about you, Christine? What's your view for the next 5 to 10 years in this field, where the big advances will take place, you reckon?
Thank you, Elena. I'll be happy to speculate. So, number one, maybe just using Thomas's comment as a segue into the question of the global view. And I think we have seen very recently through the discovery, for example, of the GBA African variant. We call it the African variant. It's a variant that is more common, or that is only found, I should say, in Africans or African admixed populations, and that is associated strongly with Parkinson's disease there. So I would predict that we will find associations, but potentially also genetic factors that are actually causative. I actually returned earlier today from the Philippines, and there's a condition, it's called X-linked dystonia Parkinsonism, which is also a form of Parkinsonism, if you will, that is endemic to a single island in the Philippines. And so I'm sure we'll discover more genetic factors. Number two, I think we will better understand, and Tanya, you alluded to this, and Thomas, you as well in your talk already, I think we will better understand penetrance. And this is a very personalized part of medicine, because as soon as somebody has their variant, and has a confirmed variant, they will ask us, and we will ask ourselves exactly this question. So when and if and how will this individual become manifest? And so I think this is one of the key questions that we will be able to answer better than we can now. And there's already also very interesting findings from the environmental field, but of course we're talking genetics today, and I'm sure we can also find more genetic modifiers if we stratify the patients well, if we take care of the, a very careful patient selection, etc. We will also have, I'm hoping, and my crystal ball kind of says that we will have in the next 5 to 10 years, we will have some treatment that actually works causally. And how well it works and whether it can halt or just modify, I don't know. But I think we will have that. And so with this, I think it will become, and genetics will help us stratify, we discussed this in detail, will help us stratify patients towards these treatments. And I think this will be another very, very important step forward. I think those are my 3 main predictions, and all are quite positive, I have to say. I hope they become true.
I hope so too, yes. Well, now I think we are eager to hear your predictions, Tanya.
Okay, now my time to speculate. I will start with 5-year horizon. Also very much optimistic, reflecting on the rapidity of the field evolution in the previous 5 years. I hope and predict that genetic testing will enter the clinic at minimum for individuals with newly diagnosed disease, both for stratification for clinical trials and for counseling regarding impact on their disease progression. In order for that to happen, it's not only individual country and region pairs understanding the significance of that, and it's our charge as academicians, as clinicians, to explain to the pairs the significance of that. We need to educate physician community so that physician community and clinicians community in general is comfortable counseling people on the reasons for the Parkinson's focused genetic testing and results, which is an essentially important part. As you've heard, it's a complex discussion, and I want to call out Christine, who is leading the charge on behalf of the Movement Disorder Society to put together recommendations for clinical genetic testing and counseling. So that's my 5-year prediction. My 10-year prediction, we will be where cancer field is today. It's genetic characterization, it's pathway characterization. I'm stepping beyond pathology. We have the biomarker of pathology today, which is synuclein biomarker. We need to do better, but pathology characterization and stepping into the pathways characterization. Those are big horizons, but the field is moving fast, and I'm optimistic.
Excellent. So I hope that our audience gets an optimistic uptake as well. Then in this bunch of questions that we have from the audience, I think there is another theme here that I will summarize as the technology theme. And these questions relate with technological advances and how genetics will move forward by the use of new technologies. And this question comes to mind then, which is I think that this is a relevant question. The fact as well is the field moving from a hypothesis-driven approach like many years ago to a non-hypothesis-driven approach in genetic technologies that require thousands and thousands of samples. So I think that geneticists are a better place to enlighten us about this, which is quite complicated, I suppose. So genome-wide, whole exome, what kind of techniques are advancing the field at present?
Well,
Perhaps
maybe Christina?
You can start, Thomas. I come next.
Okay, I can start. Well, I mean, I think there are 2 major technological developments that I can see. One is the actual data generation, so basically the sequencing, and that does not end in just whole genome sequencing, which is really a common technology now. So we're moving also in the GP2 collaboration. We're moving from genotyping, so just identifying individual variants across the genome, to whole genome sequences and whole exome sequences, and in the next step, whole genome sequences very fast. It's also a lot about other omics technologies that develop very fast and that have to develop in parallel. Because as I mentioned, there's only one step from the gene to the mRNA, which is pretty much unidirectional. Everything else is in feedback loops. So there needs to be parallel developments, and there are parallel developments in spatial transcriptomics, for example, in seeing which genes are expressed in which cells and in which cell context. There are metabolomics and proteomics that need to be developed also on a cell-specific way. So there's a lot to be developed, but I think this is what we're seeing. That's one part, the generation of the data of a very, very complex system like the human brain, most complex system in the universe, I would say. But then there's also the data processing. So all the computational aspects that are involved with that and that have to develop in parallel. So I think we're just basically at the start of a breathtaking development in those areas that will allow us much more insight than we had so far.
Excellent. Then there is another topic emerging, which, sorry, Christine, maybe I interrupted you.
No, sorry. If I may, I just wanted to add, I'm in full agreement, obviously, but just one further aspect, Thomas, I think we're moving also from whole genome sequencing short read to long read. And I think one very exciting discovery that was made last year, for example, and I'm sure there's more to come, and potentially also in the field of PD, Parkinson's disease, is, for example, FGF14 related ataxia. And this is also a late onset condition where it was found that a long repeat in the introns of more than 250, 300 repeats is actually causative of a disease that starts in the 70s. So this came as a surprise. We never even tested these patients in the clinic anymore. Now we do, and it accounts for a considerable percentage. So I'm hopeful that also even with genome-based methods, we'll still be able to uncover additional causes or risk factors, or potentially also protective factors for Parkinson's.
Right. Yeah. I was about to ask about protective factors or genetic modifiers, of course. Yeah, because I mean, in that balance of non-hypothesis-driven versus hypothesis-driven research on how, and of course we didn't talk about epigenetics, right? But I suppose that a lot is going on in that field as well, right?
Absolutely.
Yeah, and this is maybe a question or a general theme of several questions, many questions actually, that perhaps are Tanya would like to respond. They relate with the difficulties and the ethical components in relation with including prodromal patients in clinical research studies, right? People that are otherwise healthy, right? They are clinically healthy, but because of the genetics, because being the carriers of a gene, because of being stratified by use of biomarkers, they might be included in research. And of course, that's an ethical conundrum for many people. And I'm sure that, Tanya, in your long experience with those studies, you have confronted many of these questions in your career.
First of all, thank you very much for posing the question. And very important question, right? And the question that the field is actively addressing, right? What specifically focusing on the ethical considerations of enrolling individuals. Now we are speaking strictly about research. It's not clinical care. So engaging individuals with a risk profile, be that genetic risk or phenotype risk, into clinical research. So again, referring to Parkinson Progression Marker Initiative study, PPMI, which is not the only one, but I believe it has and is paving the way. Why do we need to do that? Let's start with why do we need to do that? Because I think that the mantra prevention is better than treatment, right? Is absolutely stands. And if we want to move into the domain of preventing the disease, we need to go all the way to the left, right? And what I mean by the left, people who are carriers and genetic domain is the best prototype to talk about, right? It is inborn factor. We don't know the penetrance of these genes. We need to define the predictors of penetrance, genetic, not genetic. The only way to do that is to study those individuals. Now and I don't think that anyone would argue, now from the rationale to the ethical considerations, a lot of research is going into that. And again, PPMI, I'm speaking about the study that I'm directly involved in, has recruited more than 2,000 individuals into that prodromal arm of the study, all of whom have been counseled and signed the informed consent that states that you are being enrolled in the study because you have one of the potential risk factors. So people are interested. And again, people are interested, and obviously it has to be done in the ethically very responsible way and in ethically compliant way, with a strong arm of counseling, education, built at every step of interaction with the individual. Christine has brought up a very important aspect and evolving aspect of research, return of the results. For a long period of time, we enrolled individuals with genetic variants, not sure whether we should disclose that information for them. Now the field has moved on to consented disclosure of the results. People want to know, and people are intelligent to ask the right questions. Again, provided that we provide the culturally, educationally appropriate information. Again, a lot of research is going into that. There are surveys, there are pre-disclosure of the results counseling, there is post-disclosure of the results counseling. So to sum up that question, very important question. The field is rapidly collecting and developing information, but no question about it. The field is going in the direction of full partnership with individuals, which means that they have the right, and it's our responsibility to responsibly communicate the data.
That's a very, very strong message, Tanya. Thank you very much for that. I'm really glad to learn that PPMI has set up the pathways like a template for everybody else to learn from here. So that's fantastic. We have very few minutes left for this discussion, and I think it will be very nice to end by asking the question that a young researcher is posing to you, Thomas. So she would like to know, what's your advice for her career? So apparently she's just started as a neurogeneticist, and she's asking for your advice on, what would you recommend?
Okay, so first of all, I would think that it's a good choice to go into neurogenetics because what we just enumerated, all the enormous advances that we are expecting. To be honest, I think you could go into a more experimental direction that would go a little bit back to your which we haven't really touched on quite enough, the hypothesis-driven research, which is still necessary and very important. because with all our big data, it is very difficult to really draw any conclusions on individual causative connections. So the experimental part is still very important, and this could be one way. The other way would be to go into a more computational direction. That would be another way where we can find out still a lot more. I think there's still a lot. You have mentioned epigenetics. You can also think of all the interactions between genetic variants. We are basically haven't really started to touch that yet. So there are so many things that can be done both experimentally and computationally, and I think both are very, very worthwhile paths to follow.
Well, so that's, yeah, a lot to think about from there, I suppose. Christine, will you have any recommendations for young researchers?
Yes. So again, I would like to congratulate everyone who decides to take on a career or at least an interest in the also the research part. I think for every clinician, this is really broadening your horizon. And even if you do this only for a period in your life, I think this is a good thing to do. It eventually benefits our patients, and it also benefits, I think, every one of us. I have rather generic recommendations. So one recommendation that I personally benefited from a lot is also to go abroad, go out of your comfort zone and seek out a lab or a clinic or a department somewhere else, in the world, and learn from them. See what you already do so well and what you could do better on the other hand. So I think this is an incredible experience. And then, of course, team up among yourselves. There are wonderful opportunities to team up with other young researchers. I think this is very powerful, and find good mentors. This is another, these are, like I said, quite generic, but we are, and again, I'm coming back to GP2, I'm sorry, and I'm heavily biased because I'm so involved with this network. But it is an opportunity when you're interested in Parkinson's genetics, really a wonderful opportunity to be part of a big network. And there is enough room, as I think we've seen throughout the discussion, for so many different pathways you may be able to predict, to pursue. And this can also include more, research questions at interfaces. For example, genomic imaging could be one. So there are so many different things. Try to find your niche and definitely do what really interests you, what you burn for, and what doesn't let you sleep at night.
Awesome. So if you have anything else to add, Tanya, on the recommendations for young researchers?
All I will add is it's an exciting time. So dive in, don't be scared, see the big picture. Research is moving more and more into the human models, again, led by the genetic discoveries. And again, seek collaborations, repeating what both Thomas and Christine have indicated. Seek collaborations, join multiple consortia that exist. They will welcome you, and again, develop expertise.
So thank you very, very much for this wonderful conversation. I had a great time talking to you. I hope that our audience enjoyed as well and found this conversation as helpful and informative as I did. And I would like to conclude by obviously, expressing my gratitude to the three of you. Thank you very, very much for your generosity with your time and your participation. And to finish, I would like to suggest to those that will like to keep up with this conversation to go to our website and check this recent content. These papers were just published yesterday on time for this webinar. A wonderful paper from Christine Klein and Christine Klein on their vision for the future of this field, and a link comment on that paper written by Thomas on his opinion about that global vision. I think these papers will be of interest for this audience, and I hope that you enjoy them as well. And with that said, thank you very, very much all. I hope I get to see you again very soon. Thank you.
Thanks a lot for having us.
Thank you.
Thank you very much.